Throughout human history, one of the most effective military strategies has been the siege.
Oh, absolutely.
Right, like you surround a castle, you cut off the supply lines, you block the roads, and you just wait.
Yeah, you just starve them out.
Exactly. It relies on this really simple, almost finite equation. A closed system that runs out of resources eventually has to surrender.
Because without fuel or food, things just have to stop. It's basic physics and biology, really.
Right. And for a long time, that was exactly how we conceptualized treating certain types of aggressive solid tumors. We thought, if we could just besiege the cancer, cut off its blood supply, we could just starve it into submission.
Yeah, and it's comforting to think of it that way, I think, because it feels predictable. You apply a block, the system fails.
But then you step into the actual microscopic world of cancer biology, specifically the microenvironment of liver cancer. And suddenly, that besieged castle, it doesn't surrender at all.
Not even a little bit.
When you cut off the supply lines, the cancer turns into this, master escape artist.
It is the absolute definition of an evolutionary arms race happening right inside the human body.
Right.
When you apply immense pressure to a tumor, you are forcing it into a binary choice, adapt or die.
Right.
And unfortunately, these cells have developed incredibly sophisticated, almost science fiction level ways to choose adaptation.
Which is just wild to think about. So welcome to the deep dive. Today, our mission is to decode one of the most complex and surprising survival strategies used by liver cancer cells.
It's a fascinating one.
It really is. We are unpacking this dense, cutting-edge research paper with a title that is so packed with jargon, it's practically a different language.
Oh, it's a mouthful.
It is. It's called, and bear with me here, CD147 positive migrasome macropinocytosis promotes HCC sorafenib resistance via inducing vasculogenic mimicry triggered by PI3-AKT twist1 signaling.
I know. It is a massive wall of alphabet soup.
Totally.
But beneath that extremely dense title is an unbelievable story. Every single one of those acronyms represents a massive breakthrough in understanding how tumors stay alive when we try to kill them.
Yeah.
And more importantly, how we might finally stop them.
Exactly. And to set the stage for you listening, we are talking about hepatocellular carcinoma, or HCC.
Right, which is a major global health issue.
Yeah. It's a very aggressive, very common form of liver cancer. And when a patient is diagnosed with advanced HCC, the first-line defense is often a drug called sorafenib.
Yes, sorafenib.
Now, sorafenib is essentially our siege army. It is a targeted therapy specifically designed to choke off the tumor's blood supply. It cuts the fuel lines.
And initially, it looks like a miracle drug. The blood vessels degrade, the tumor is starved, and the patient scans look great.
But there is a massive clinical hurdle that doctors face.
Yeah, a huge one. Tumors very often develop something called Sorafenib Resistance, or SFR. The drug just mysteriously stops working.
Like, completely.
Pretty much. The tumor just starts growing again, totally unfazed by the siege.
Which is exactly what this source material dives into. Today, we are going to explore how these cancer cells manage to outsmart our best drugs by essentially leaving these microscopic survival care packages for each other.
Yeah, and to really grasp the mechanics of this, we have to start at the exact moment the drug starts working.
Okay, take us there.
So when sorafenib successfully chokes off the blood supply to the tumor, it creates an environment of severe low oxygen and low nutrient stress. In clinical biology, we call this state hypoxia.
Right, so going back to our siege analogy, the drug has surrounded the castle. There is zero oxygen getting in.
None.
The glucose is completely gone. The cells inside are quite literally suffocating. So what exactly happens in that moment of crisis?
Well, they panic.
Yeah.
But it is a highly organized biological panic.
An organized panic, I like that.
Yeah. The researchers found that under this extreme hypoxic stress, the cancer cells that are trying to migrate away from the dead zone, they start leaving something highly specific behind.
What are they leaving?
We're talking about these newly discovered large extracellular vesicles called migrasomes.
Okay, let's unpack this for a second.
Yeah.
Because migrasomes are a relatively new concept in cell biology, and they are fascinating.
They really are.
When we say large in the cellular world, we mean about 0.5 to 3 micrometers in diameter, which for a cell is pretty chunky.
It's massive for a vesicle.
Right. So picture a cancer cell fleeing from that toxic, suffocating environment. As it crawls away, it literally pinches off intact pieces of its own cell membrane, just leaving them behind on the tissue like a trail of breadcrumbs.
That's a great visual. But these aren't just empty fragments, or cellular trash. They are meticulously packed survival kits.
Survival kits.
Exactly. And this is where the paper gets very specific. Under normal, healthy conditions, a migrating cell might leave a few migrasomes behind just as a byproduct of moving.
Just normal wear and tear.
Right. But under the combination of extreme hypoxia and low glucose, that suffocating siege we talked about, the HCC cells start pumping out these migrasomes in massive, desperate quantities.
Like a factory in overdrive.
Exactly. And the researchers call these specific stress-induced packages hypomigs.
Hypomigs. Okay. And the most important part of these hypomigs isn't just that there are a ton of them, it's what they're wearing on the outside.
Right. The outfit matters.
Right, because the researchers discovered that these specific stress-induced migrasomes are heavily coated in a glycoprotein called CD147.
Precisely. CD147 is a transmembrane protein, meaning it sticks out on the surface of the migrosome like a little flag.
Okay.
And in oncology, CD147 is notorious. It's well known for promoting multi-drug resistance.
Oh, wow.
Yeah, so the hypoxic stress essentially forces the cell to not only manufacture more of these survival kits, but to deliberately coat them in this CD147 armor.
It's like the molecular stamp on the outside of the care package that says, urgent survival instructions inside.
That is exactly what it is.
And what I found so compelling about the source material is that the researchers didn't just observe this in some isolated petri dish in a lab.
No, they took it much further.
They actually went to massive patient databases and tissue microarrays to see if this was happening in real human beings treated with sorafenib.
They did. And the clinical correlation they found is incredibly stark.
What did they find?
Well, they analyzed the tissue of real HCC patients and looked for two things. First, high levels of hypoxia, which they track using a marker protein called HIF-1 alpha.
HIF-1 alpha. Okay, that's a suffocation marker.
Exactly. And second, high levels of specific proteins that indicate migrasome production. These have names like ITGA5, TSPAN4, and EOGT.
Which, just to translate that for you listening, basically means they were looking for tumors that were severely suffocating and at the very same time dumping out tons of these breadcrumb care packages.
Right.
And what did that mean for the actual patients?
It directly correlated with significantly shorter overall survival.
Wow.
Yeah. If a patient's tumor was highly hypoxic and producing a high volume of these migrasomes, the sorafenib failed much faster.
It just stopped working.
Right. So the clinical data proved that this isn't just an interesting cellular quirk. These migrasomes are a primary real-world driver of drug resistance.
Okay, so let's do a quick recap before we move on to the next phase, just to lock this in.
Sure.
So for you listening, here's where we are. The drug sorafenib besieges the tumor. The tumor experiences severe hypoxia, which we track with HIF-1 alpha.
Right.
The dying cells panic and drop a massive amount of survival care packages called hypomigs, marked by things like ITGA5 and TSPAN4. And crucially, these packages are heavily armored with the CD147 protein.
Perfect summary.
Awesome. So my question to you, the listener, as we go into this next section is this: how on earth does a starving cell actually eat a giant care package?
Yeah, that's the big mechanical question.
Right. Because it's one thing for a fleeing cell to drop a heavily armored package on the ground, but how does the neighboring starving cancer cell actually pick it up and use it? I'd assume swallowing a massive foreign object would trigger some kind of cellular indigestion or, I don't know, even kill a cell that's already starving.
It's a great point. And that is the exact crucial transition we have to look at. Going from the release of the migrasome to its absorption by a neighbor.
Okay, so how do they do it?
The mechanism the researchers uncovered is a process called macropinocytosis.
Macropinocytosis. Okay, and to visualize this, you have to think about how cells usually take in nutrients, right?
Yeah, normally it's very polite.
Polite, exactly. Highly regulated and specific, like a single molecule binds to a specific receptor on the surface, and the cell delicately pulls it in.
Right. Receptor-mediated endocytosis.
Yeah, it's like sipping a drink politely through a tiny, precisely engineered straw.
But macropinocytosis, that is not polite at all.
Not at all. The prefix macro means large, and pino means drinking.
Bulk drinking.
Literally. The starving cancer cell literally folds its outer membrane outward like a giant pair of lips and takes a massive, indiscriminate gulp of the fluid around it.
It is literal bulk drinking, yeah. The cell is so starved of glucose and oxygen that it desperately swallows huge volumes of everything in its immediate environment.
Just hoping to catch something useful.
Exactly, just hoping to pull in some ambient nutrients to survive.
Yeah.
And in doing so, it inadvertently, or perhaps purposefully swallows those large CD147-coated migrasomes that the fleeing cells left behind.
But the researchers found that this massive gulping action isn't happening entirely by accident, right? Like, the cell doesn't just spontaneously start drinking the ocean. There is a specific trigger.
Yeah, what's fascinating here is that they identified the exact molecular key to this lock.
What's the key?
It's a receptor on the surface of the cell called LPAR6. That stands for lysophosphatidic acid receptor 6.
LPIR6.
Right. The presence of this specific receptor acts as a sensory trigger. When LPR6 is active, it basically permits the HCC cells to perform this massive gulping action to take in the migrasomes.
And the way they proved this is just brilliant. It's a classic scientific proof by subtraction.
Oh, yeah, the knockdown experiment.
Right. I mean, if you want to know if a specific spark plug makes an engine run, you take it out and see if the car stops. So the researchers performed what's called a genetic knockdown experiment. They went into the DNA of the HCC cells and literally silenced the gene that manufactures LPR6.
And the results were immediate and profound. When LPR6 was knocked down, the cells completely lost the ability to perform macropinocytosis.
They just stopped drinking.
They stopped drinking. And because of that, the CD147 migrasomes just sat there in the extracellular space, completely unabsorbed.
Just floating around outside the cell.
Yeah, useless.
And because they couldn't absorb those survival care packages, the sorafenib drug suddenly started working again.
Yep.
The cancer cells trapped in the siege without their survival instructions were pushed back into apoptosis, which is the biological term for programmed cell death.
Right. So they died.
Right. So just blocking the gulping mechanism restored the drug's lethal power.
Exactly. The survival instructions are out there, but they only work if the neighboring cell can actually get them inside its own walls. The cancer cells use macropinocytosis to swallow them, and this is entirely dependent on that LPIR6 receptor acting as the watchtower or the trigger.
Okay, time for another quick mini recap.
Let's do it.
So step 1, panic and drop CD147 packages. Step 2, which we just covered, the neighboring cell uses the LPR6 receptor to trigger macropinocytosis, this massive gulping, to swallow the package whole.
That's the sequence.
So my next question for the listener is, once that package is inside, what do the instructions actually say?
Oh, this is arguably the most mind-blowing phase of the whole process.
It really is. Let's say the cell does have LPR6. It takes a massive gulp. The CD147-coded care package is now inside the neighboring starving cell. What exactly are the instructions inside? Because here's where it gets really interesting. Reading this message literally changes the fundamental identity of the tumor.
It does. To understand this, we have to look at the biological chain reaction that happens the moment that CD147 migrasome is broken down inside the new cell.
Okay, walk us through it.
It activates a very specific signaling cascade. So the CD147 triggers a kinase enzyme called PI3K.
PI3K.
Right. PI3K then activates another protein called AKT.
AKT.
And AKP ultimately wakes up a transcription factor called twist1.
Okay, PI3K to AKT to twist1. We are firmly back in alphabet soup territory here.
Yeah, we are.
Break that down for us because it sounds like a secret military chain of command.
That is exactly the right way to think about it. It is a strict chain of command. I like to use a mnemonic here. Think of PAT.
Okay, I like that.
P is for PI3K. Think of PI3K as the general at the perimeter of the cell who receives the initial message from the migrasome.
The perimeter general, P, got it.
Right. The general hands the orders to the active captain, AKT, that's the A.
Active captain A.
And then AKT runs into the headquarters, the nucleus of the cell, and hands the final execution orders to twist1, that's the T.
The T twist1. And twist1 is a transcription factor, right?
Exactly. It's a master regulator, essentially the chief engineer. When twist1 gets activated, it starts rapidly changing which genes in the cell's DNA are turned on and off.
Which is a huge deal.
It's massive. It tells the cell to change its physical shape and its entire biological function.
So what does this all mean for the actual behavior of the tumor? Let's go back to the castle analogy. The drug destroyed all the normal blood vessels.
Right.
The tumor is suffocating. The cell reads these engineering instructions from the migrasome via the PAT chain of command. And then what does it actually build?
It builds its own DIY plumbing system.
DIY plumbing.
Literally. The biological term for this process is vasculogenic mimicry, or VM. Because the sorafenib wiped out the normal, healthy blood supply, the cancer cells use the twist1 signal to physically mutate. They undergo a massive structural transformation.
I have to say, this part of the source material genuinely blew my mind. I need to push back on this for a second because it sounds like pure science fiction.
It really does.
Are you saying a liver cancer cell just, becomes a blood vessel. How does a cell completely change its biological identity like that?
It is incredible, but yes, they literally shapeshift. It's a process related to something called epithelial to mesenchymal transition, or EMT.
EMT, okay, explain that.
Normally, liver cancer cells have epithelial markers. These are surface proteins that make them act like solid, stationary organ tissue.
Okay.
Specifically, they have a protein called E-carvin.
E-chi.
Think of E-carvin as biological mortar. It keeps them locked in place, stuck together as solid liver cells.
Right.
But under the command of Twist 1, the engineer, they shed that E-carvin. They completely lose their normal organ identity.
They drop the mortar.
Exactly. They drop the mortar. And in its place, they generate entirely new tools. They start expressing markers like VE-cotterin and enzymes called MMP2 and MMP9.
Okay, VE-cotterin, MMP2, MMP9.
Yeah, you can think of these new proteins as microscopic scissors and sticky Velcro.
Scissors and Velcro. Okay, I can picture that. These are markers typically only found on vascular endothelial cells, these specialized cells that make up real veins and arteries. By generating these scissors, they can cut through the surrounding tissue.
Wow.
And by generating the Velcro, they gain the ability to physically link together, stretch out, and form hollow tubes.
Which is just terrifying. They form fake, rogue blood vessels.
Yes, vasculogenic mimicry.
There aren't real veins. They aren't real arteries. They are hidden underground tunnels built entirely out of mutated liver cancer cells.
Exactly.
And this allows ambient blood and nutrients to flow directly into the dead center of the tumor.
Yep.
The cancer is literally digging its own secret ventilation shafts and supply lines to survive the siege.
It's a terrifyingly elegant solution to the hypoxia problem. But of course, the researchers needed a way to prove this was actually happening.
Right, because if you're looking at a tumor under a microscope, a tube is a tube.
Exactly.
How do you spot the fakes? How do they prove these are constructed out of mutated cancer cells, not just normal blood vessel cells that somehow survived the drug? They used very specific cellular stains to identify them.
Okay.
Normal, healthy blood vessels always express a marker called CD31. So if you stain for CD31, normal vessels light up.
Okay.
But these rogue cancer-built vessels do not. They are CD31 negative.
Oh.
However, because they are acting like vessels and forming tubes, they do manage to create a structural basement membrane. And that shows up positive for a completely different stain called PAS.
PAS, so they are PAS positive.
Exactly.
Yeah.
So the researchers scan the tissue looking for structures that are PAS positive, but CD31 negative.
That's brilliant.
When they see that specific combination, they know without a doubt they're looking at vasculogenic mimicry. They're looking directly at the tumor's secret plumbing.
Okay, that is a lot of incredible biology, so let's just make sure we've got this entire chain of events locked in with our final mini recap here.
Yeah.
Because this sequence is the absolute core at how the resistance works.
Let's do it.
All right, so you're listening. Picture this. The tumor gets besieged by sorafenib and starts starving. Right. The fleeing cells panic and drop heavily armored, CD147-coded migrasomes.
The care packages.
The neighboring starving cells gulp those packages down using bulk drinking or macropinocytosis, which is triggered by the LPR6 watchtower receptor.
Right on.
Once the package is inside, the CD147 triggers the PAR-T chain of command. The PI3K general tells the AKT captain, who wakes up the Twist 1 engineer.
Yep.
And finally, Twist 1 tells the liver cell to drop its normal mortar, grow some microscopic scissors and Velcro, and build fake blood vessels so the tumor can breathe again.
That is the complete multi-stage blueprint of how hepatocellular carcinoma beats our best drug. It is a highly coordinated, flawlessly executed survival playbook.
Which poses the ultimate therapeutic challenge for you, the listener, to consider. If scientists have finally decoded this entire playbook step by step, how can they use it against the cancer?
Yeah, how do you break it?
Right. How do you tear up the blueprints?
I'll answer that. The researchers moved out of the Petri dish and into in vivo testing. They used what are called CDX models.
CDX models, what are those?
These are live mice that were injected with human liver cancer cells. So once the human tumors grew inside the mice, they started feeding them sorafenib to initiate the siege.
And just to prove the mechanism was real, the researchers actually took those CD147 positive hypomigs they harvested earlier and injected them directly into the mice alongside the drug.
They did.
And the result was exactly what you'd expect based on the playbook, right?
Oh, yeah. It was dramatic. The tumors grew wildly. The migrasomes completely overrode the sorafenib.
The drug was utterly useless.
Completely useless, because the cells immediately used the migrasomes to build their fake blood vessels.
But then came the true intervention, the counterattack.
Right. If we connect this to the bigger picture of oncology, treating a complex, highly adaptive system like a tumor often requires more than just one weapon.
You can't just rely on the siege.
Exactly. You need a multi-pronged approach. So the researchers designed a two-pronged attack to dismantle this specific resistance mechanism at two entirely different vulnerable points.
Okay, break down the two prongs for us.
They introduced two specific inhibitors into the mix alongside the sorafenib. First, they used a compound called wortmannin, or WTM.
Wortmannin. And what does wortmannin do?
It specifically inhibits that first general in the PIT chain of command, the PI3K pathway.
The perimeter general. Okay.
Right. And remember, PI3K isn't just about sending messages to the nucleus. It's also a major driver for the macropinocytosis itself, the bulk drinking.
Oh, so by applying wortmannin, they're essentially taping the cancer cell's mouth shut.
Exactly. Even though the survival care packages are floating all around them in the environment, the cells simply can no longer gulp them down.
That is genius. But they didn't stop there, right?
No, because as we've seen, cancer is incredibly tricky and redundant.
Right. I mean, if even one package gets through the tape, the tunnels get built.
Precisely. So they added a second prong, a drug called polyphilin I, or PPI. And in some of their tests, they also used a drug called doxycycline, or DOX.
Okay, polyphilin I and doxycycline, what do they do?
These drugs target the very end of the signaling cascade. They suppress twist 1, the chief engineer.
Wow. So consider the elegance of this dual blockade. Even if a few migrasomes somehow manage to slip inside the cell past the wortmannin, suppressing twist 1 means the cell simply cannot act on the instructions.
Exactly. It stops the cells from mutating their surface markers. It completely halts the generation of the scissors and the Velcro, meaning the construction of the fake blood vessels is completely shut down.
It's brilliant. You block the gulping at the membrane and you burn the blueprints in the nucleus.
It's a complete structural shutdown.
And the climax of this entire massive experiment was just undeniable. When they applied this heavy-hitting combination, sorafenib plus wortmannin plus polyphilin, it absolutely crushed the tumor's resistance.
The microscope slides from the mouse models were definitive. Those PAS-positive, CD31-negative fake blood vessels, they practically vanished from the tissue samples.
They just disappeared.
Yet, without their DIY plumbing, and without the ability to share survival instructions with their neighbors, the cancer cells were left highly vulnerable. The siege held.
That's incredible.
Sorafenib regained its full lethal efficacy against the tumor, driving the cells rapidly back into apoptosis.
And there's a really crucial detail to note here for any future clinical application in humans. Because we always have to think about toxicity, right?
Always.
This combination therapy did all of this without causing body weight loss in the mice.
Which is a huge deal.
Right. In drug testing, severe weight loss is a major indicator of toxic side effects. So this wasn't just carpet bombing the animal. It was a targeted, tolerable, and highly effective dismantling of the cancer's specific defense system.
It perfectly illustrates why we have to understand the microscopic mechanics of the tumor microenvironment on this level.
Yeah.
You can't just throw a targeted drug at a tumor, assume the vault is locked, and expect the cancer to just sit there and take it.
Right. You have to anticipate its evolutionary countermoves and cut them off.
Exactly.
So, bringing this incredible journey to a close, we started with a drug that seemed to mysteriously fail, but by zooming way, way in, researchers decoded this epic microscopic battle.
An absolute war zone.
The siege drug causes hypoxia, the suffocating tumor panics and drops armored migrasomes. The neighbors gulp them down, activating a chain of command that tells the cells to literally shapeshift and build fake blood vessels to survive.
Yep.
And the way we finally beat it is by taping the cell's mouth shut with wortmannin to stop the gulping and burning the blueprints with polyphilin to stop the building.
Which is an amazing victory for science. But, this raises an important question, though.
Oh, what's that?
Well, we've just spent all this time decoding one specific pathway using one specific vesicle in one specific type of cancer.
Right.
But if we now know that cancer cells actively use these bulk drinking mechanisms to indiscriminately absorb complex instructions from their environment, could scientists eventually hack this system?
Oh, wow. That is a fascinating thought. You're talking about flipping the script.
Exactly. If the cancer cell is indiscriminately gulping down packages, it assumes our survival instructions. Could we one day pack our own synthetic migrasomes.
Like a Trojan horse.
Exactly. Could we create Trojan horse packages filled with instructions that tell the tumor to destroy itself and just let the cancer's own desperate bulk drinking do all the delivery work for us?
That is the ultimate biological espionage.
Isn't it?
I love that. The true victory against cancer might not just be locking the gates of the castle. It might be intercepting their secret messages, learning their microscopic language, and eventually sending our own orders right into their headquarters.
I think that's the future of oncology, honestly.
What a thought to end on. Thank you for joining us on this deep dive. Stay curious, because as we've seen today, asking exactly how something survives is always the best way to decode the world.